Hello, everyone, and welcome to today's webinar, The Basics of Protein Analysis. Topic 1, Fundamental Antibodies for Applied Research. Topic 2, Practical Considerations for Quantitations and Western Blotting. I am Megan Pascal of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you're having trouble seeing or hearing the presentation. This webinar is also equipped with multiple resources found in the resource window on the right side of your screen. Here you can download PDFs, visit relevant URLs, and even download today's PowerPoint presentation. I'd like to now welcome our speakers, Dr. Audrey Lim, Product Manager, SEA, and Dr. Anthony Lim, Technical Sales Leader. Dr. Audrey and Anthony Lim, you may now begin your presentation.
All right, thanks, Megan. Good afternoon, everybody. Today, Audrey and myself will be sharing on the topics of antibody for the applied research. So I'll start by kicking off on the fundamentals of the antibodies. This is the outline of my presentation today, where I'll start with an introduction to the antibodies, the different classes and clonality of antibodies. Then I'll follow up by sharing some of the antibody-based technique widely used in the research field. This slide describes the history of antibody development. In the early 1990s, Paul Ehrlich, who proposed the side chain theory, where he hypothesized that side chain receptors on cells bind to a given pathogen. He was the first to propose a model for an antibody molecule in which the antibody was branched and consisted of multiple sites for binding to the foreign materials known as an antigen. For the activation and the complement pathway, Behring was then awarded the Nobel Prize in the 1901 for his work on the serum therapy. Later part of the year, antibody engineering technology was introduced in the early 1990s, where then it actually led to the application of the antibodies as important biopharmaceutical products. Each antibody recognizes one specific antigen. For example, an antibody that recognizes the mumps virus cannot recognize the measles virus. Conversely, an antibody that recognizes the measles virus cannot recognize the mumps virus. This feature is known as antibody specificity. Each B cell, or also known as antibody-producing cells, is able to produce one kind of antibody. Antibody have different functionality. Firstly, on neutralization, where the neutralizing antibody are known to bind to viruses as well as other particles. Later part of the year, the complement system was introduced as part of the immune system that enhances or complements the ability of antibodies and phagocytic cells to clear the microbes and also damage cells from an organism which then promotes inflammation, as well as they attack the pathogen cell's membrane. Phagocytosis is a cellular process for ingesting and eliminating particles such as microorganisms, foreign substances, as well as apoptotic cells. Antibody affinity and avidity. So affinity is defined as the strength produced by a single epitope interaction with a paratope on the antibody. Antibodies require a high specificity towards its epitopes, and this is commonly known as low strength. Avidity thereby refers to the accumulated strength of multiple affinities of an individual non-covalent binding interaction, such as between a protein receptor and its ligand. Here shows some of the monomeric or divalent antibodies, including the IgG, IgD, as well as the IgE. There are also the multivalent antibodies, which includes the IgM, as well as the IgA. Monoclonal antibodies have monovalent affinity, binding only to the same epitope. In contrast, polyclonal antibodies bind to the multiple epitopes and are usually made up of by several different antibodies secreting plasma cells lineages. Bispecific monoclonal antibodies can also be engineered by increasing the therapeutic targets of one monoclonal antibody to 2 epitopes. Some of the features of monoclonal antibodies may include identical lots. Monoclonal antibody recognizes only one particular protein form, and they are commonly known to be expensive to produce. On the contrary, polyclonal antibodies display a lot-to-lot variability. They are tolerant to small changes in protein structures, and commonly they are a lot more economical to the cost of production. Antibodies are generated by immunizing host animal with an immunogenic substance. Immunogen can be full-length protein, protein fragments, peptides, whole organisms such as a bacteria or even cells. The immunogen of the antibody will define which region of protein antibody binds to. Monoclonal antibodies are typically made using B cells from an immunized animal which are fused to a immortalized cell line to form a hybridoma that secretes the desired antibody clone. This hybridoma technique produces highly consistent, specific, as well as sensitive monoclonal antibodies in large quantity. Recombinant antibodies are produced in vitro by cloning antibodies gene from an immune-specific heavy and light antibody chain into high-yield expression vectors. These vectors are then introduced into expression hosts such as bacteria, yeast, or even mammalian cells to generate the recombinant monoclonal antibodies. The detection of antibodies can be done either by a direct method or an indirect detection method. Direct detection will include the use of the primary antibody that's directly conjugated to a label, for example, a fluorophore, and this can be used in flow cytometry techniques, which is shown in the figure here. Indirect detection, the primary antibody is bound to a labeled secondary antibody that has been raised against the host species of the primary antibody. Indirect methods may also include the amplification step to increase the signal intensity In this case here, it's more commonly used in applications such as Western blotting, ELISAs, immunocytochemistry, immunohistochemistry, as well as flow cytometry. The choice of using a direct or indirect detection is often prescribed by the expression level of the target antigen. To visualize the presence of the target protein, secondary antibodies are conjugated to fluorescent dyes or protein or the enzymes that are able to induce an enzymatic reaction to produce some light for detection. Fluorescent labels emit light in the visual range when excited by light of a specific wavelength. There are several available, all with their own excitation and emission characteristics. Immunoassays are used commonly to detect the presence of a specific protein or antigen in cells or even tissues by using a specific antibody against a target of interest. This permits downstream scientific analysis using methodology such as flow cytometry, immunofluorescence, as well as immunohistochemistry. Immunoblotting, which is shown on the right, or commonly known as Western blotting, uses the transfer of biological samples onto a membrane that permits the detection of specific antigen or protein samples using a specific antibody against the target. Our next presenter, Dr. Audrey Lim, will be sharing further insights into this technique. Shown here are some of the common applications that are used, which includes, firstly, immunoprecipitation, or IP for short. This is a technique whereby small-scale affinity purification of the antigen using a specific antibody that is immobilized onto a solid support, such as a magnetic particle or a agarose resin. It is used to study both protein-protein interaction as well as protein-DNA interaction. Immunoprecipitation was first developed as an adaptation of the traditional column affinity chromatography, with the idea to isolate just enough protein or DNA to be able to measure it by Western blotting or other semi-quantitative or quantitative assays method. An antibody, whether it's going to be monoclonal or polyclonal against a specific protein, is pre-immobilized onto an insoluble support, such as the agarose or the magnetic beads, and then incubated with the cellular lysate containing the target protein. During the incubation, gentle agitation of the lysate allows the target protein to bind to the immobilized antibody. The immobilized immune complexes are then collected from the lysate, eluted from the support, and analyzed based on the nature of the target antigen. So within Thermo Fisher Scientific, we have actually came out with this term called advanced verification tech, whereby antibodies are extensively tested in our laboratory to confirm the antibody specificity in several experimental setups. These include relative expression of the target in tissues or cell lines model, enhanced or reduced expression of the target using chemical treatment, siRNA-mediated knockdown of the protein. These experiments are developed to justify the quality of the antibody for our customers and to enhance the customer's experience. Shown here are some of the testing methods that we use commonly to verify the specificity of our antibodies. These include knockout, knockdown, cell treatment, and relative expression testing. We hope that you are now better equipped with the tools to take your research to the next level. Thermo Fisher Scientific has a large range of antibodies for you to select an antibody that suits your need for your research requirements. With this, I'd like to then acknowledge my colleagues from the North America as well as the India team who had helped to prepare these materials that I just presented. So with this, thank you, and I will then hand over my time over to Dr. Audrey Lim for the next part of the presentation.
Thank you, Anthony. Good afternoon, everyone. My name is Audrey, and I'm the product manager covering the protein analysis portfolio in the Southeast Asia region. And so today, for the second topic, I will be taking you through practical considerations for quantitation in Western blotting. Now, along with ELISAs, Western blotting is still one of the most widely performed protein detection applications, and an estimated of 5 to 8 million Western blots are performed globally each year. So why do people perform Western blotting? Traditional Western blotting is useful for detecting the presence or absence of a specific protein in a lysate or complex mixture. And furthermore, it can provide a qualitative and also quantitative assessment of changes in protein expression levels in response to stimuli or interventions. Western blotting has become essential, but it is still not loved. Ideally, we want to achieve the blot on the left, which has high signal intensity with a linear response to increasing levels of the target protein. However, more often than not, we may get saturated non-linear signals that are not quantifiable, or worse still, blots that look very blotchy or bands that are burnt out. So today, our focus will be on addressing these issues so that we can get accurate data for quantitative reporting. The problem with trying to get accurate quantitative data from Western blots is many factors can affect the quantitative results. Experimental variations, inconsistent sample and target concentrations, unequal sample loading, uneven protein transfer, saturated immunoprobing, and poor detection can give you misleading results. So in order to tell if a variation in expression is real, experimental design, controls, and normalization needs to And every step in the Western blotting workflow is important, from the separation to the transfer and to the detection. At Thermo Fisher Scientific, we provide a wide product portfolio for your Western blotting needs that are designed to save you time and improve your results. Some of our products that will transform your Western blotting experience include a reliable precast gel electrophoresis system, rapid 7-minute transfer systems, validated antibodies, automated blot processes, high sensitivity substrates, and our eye-bright imager that supports multiplex detection and quantitation. Even before beginning the Western blotting process, it is important to consider how you are preparing your protein sample. Common sample preparation issues include excess salt, high detergent concentration, incorrect lysis buffer concentration, DNA contamination, incorrect protein load amount, and excess or incorrect reducing agent. Of these issues, the most common problem for accurate quantification is using excess protein load amount. Let's see what happens with the signal intensity when too much protein is loaded. In the figure on the right, the dotted line represents the ideal situation where the relative intensity of a protein band shows a linear response with increasing protein load. And you can see this nice linear increase in red when we stain for total protein using Thermo Fisher Scientific's no-stain reagent. However, when we look at some of the commonly used housekeeping proteins like actin, GAPDH, and tubulin, you can see that the signal starts to lose its linearity at the higher load amount. From the housekeeping proteins, then your in this figure, we have performed Western blotting for actin in HeLa lysates, and from left to right, we have used varying concentrations of primary and secondary antibodies. As you can see in the corresponding graphs below the blots, antibody concentration has minimal impact on the curve. And this means that no matter how much you optimize your antibody concentrations, it will have little improvement to the linearity if the membrane is already saturated due to overloading of proteins. So, for a good Western blot experiment, it is always important to bear in mind the sample load amount. The amount of protein you load will influence everything downstream, including your transfer results, antibody concentrations to use, and also the detection reagent to choose. Too much protein can cause some of the excess to blow through the membrane during transfer or fail to bind to the membrane, affecting the accuracy of your data. During immunodetection, too much protein can cause protein bound to the membrane to be buried and not accessible to the antibodies. So, limit your sample load amount based on abundance of your protein of interest. High abundance proteins tend to saturate with loads as low as 1 to 3 micrograms, while low abundance proteins may be linear with up to 40 micrograms of lysate. For most Western blots, we recommend loading 1 to 10 micrograms of lysate, and for low abundance proteins, increasing to 5 to 30 micrograms may be appropriate but not required. Another consideration for quantitation and linearity of Western blot results is primary and secondary antibody concentrations used. Too much primary can cause non-linear antibody binding due to interference. And too much primary or secondary antibody can cause high background, which limits your limit of detection. Too much secondary antibody can result in substrate burnout or non-linear light production due to the enzyme kinetics. Too little secondary can reduce the dynamic range by limiting the limit of detection or result in excessive exposure times required. Recommended antibody dilution from vendors are just general guidelines, and often better results can occur outside of the recommended range substrate used. As such, antibody concentration optimization is important to ensure proper signal. Higher concentration of antibody does not always mean better results, even when working with low abundance targets. Too much secondary can quickly saturate your signal, especially with high sensitivity substrates. On the right, you can see some undesirable effects that may be caused by using the incorrect antibody concentrations. In the first blot at the top, the very high background is likely because too much primary or secondary antibody was used, or exposure time was too long, or a weak membrane blocker was used. On the other hand, too little antibodies used, coupled with a poor sensitivity substrate, will result in faint, undetectable bands, as you can see in the second blot. In the third blot, you can see saturated bands that are burned out, likely because too much secondary antibody was used together with a fast substrate. And in the last blot at the bottom, there are many non-specific bands, which could be due to poor antibody specificity, too much primary antibody, or insufficient blocking. To further understand the importance of optimizing antibody concentration, here we have performed Western blotting for HSP90, a high abundance target, and used different primary antibody amounts. For all the primary antibody concentrations, the curve bends due to membrane saturation. But also notice that when less antibody is used, in the line in green, the curve bending is less pronounced. And this is because antibody binding interference is avoided. And when antibody concentration is very high, like the line in blue, there is even a drop in signal due to substrate burnout because the substrate amount cannot keep up with the kinetic of the high levels of HRP present. Now let's take a look at cyclin B, a low abundance target. Here, the slight curve in data A is likely not due to overloading since it is a low abundance target. In data B, we have diluted primary antibody by further 10 times, and in data C, we have diluted the secondary antibody by further 5 times. And for both of these dilutions, you can see there is improvement in the linearity of the curve. When we diluted both the primary and secondary antibody together, the linearity of the curve was even better. And this shows that adjusting both primary and secondary antibody amounts results in near perfectly linear results. However, take note that diluting your antibodies excessively can result in minor reduction in the limit of detection for some other targets. Here, we have data showing increasing dilution of primary antibody for P23 from left to right, and in each figure, you can see 2 dilutions of secondary antibody in blue and yellow. You can see that with more diluted secondary antibody, the R-square is improved. However, when you look at the corresponding graphs below, which zoom in on the lowest target amounts, you notice there is a minor reduction in the limit of detection. Another tip is non-proportional ratios of primary and secondary antibody can often have good outcomes. So don't just dilute your secondary antibody. If you are using high primary, consider using low secondary. And if using low primary, consider increasing your secondary antibody. One common question with antibodies is, should we be reusing the antibodies? Generally, reusing antibodies is okay. However, you need to bear in mind that the results will change with each blot. As you can see from the plots and the graph, the signal gets weaker each time the antibody is reused. And so this is not ideal if you're trying to optimize your blots. Our recommendation is to start with a higher dilution of the primary antibody and use it only once. Apart from optimizing antibody concentration, choosing the right products for immunoprobing can affect your Western blot results. Thermo Fisher Scientific offers a wide portfolio of products for immunoprobing in Western blot detection, including a wide variety of blocking buffers, primary and secondary antibodies validated for Western blotting, and we also offer a full range of substrates. When it comes to detection, traditional chemiluminescence methods are very widely used and is very sensitive, but for this method, the number of targets you can visualize on a blot is more limited. Fluorescence-based Western blotting allows you to multiplex several targets on the same blot without the need to strip and reprobe. As such, fluorescence Western blotting can be used to provide more insight into complex research questions. Multiplexing is very useful when looking at targets that have the same or are very close in molecular weight, such as in the figure on the right. You can see that total MAC1 and phospho-MAC1, they have the same molecular weight, and the housekeeping protein GAPDH, which is also very close in molecular weight. And with fluorescence Western blotting, it is possible to visualize all of these on the same blot. It is also possible to combine chemi and fluorescence detection on the same blot. Next, do substrate kinetics impact quantitation? In the blots on the right, we allowed blots for Ras10, a low abundance protein, and HSP90, a high abundance protein, to sit in a small amount of substrate for extended times. For both low and high abundance bands, the data remains linear over time. However, with overloaded high abundance targets, extended incubation can cause the substrate to burn out over time. Therefore, it is important to use the substrate with the right kinetics depending on the abundance of your protein. At Thermo Fisher Scientific, we offer a range of substrates from the lowest sensitivity on the left to the highest sensitivity on the right. So you can see that our Pierce ECL is the most economical for high abundance proteins. Our Super Signal West Pico Plus substrate is an excellent everyday substrate. At the mid-range, we have Super Signal West Dura, which provides long-lasting signal duration and more flexibility for both high and low abundance substrates. And for high sensitivity substrates, we have the Super Signal West Femto, and our latest addition to the range is our Super Signal West Atto substrate, which gives you the higher signal-to-noise ratio. Here, we have benchmarked how Super Signal West Dura, Pierce ECL, and Super Signal West Atto performs with different primary and secondary antibody concentrations on Ras10, a low abundance protein. The results show that at high antibody concentrations, Super Signal West Atto is way too sensitive, showing a non-linear curve and signs of burnout. On the other hand, when we use the lowest sensitivity option, which is the Pierce ECL, this resulted in exposure times that were way too long in the low antibody concentrations. What we notice, however, is that for Super Signal West Dura, which has a mid-range sensitivity, it actually performs very well across different antibody concentrations, showing a linear curve and fast exposure times. Interestingly, even with high abundance proteins like HSP90, Super Signal West Dura substrate performs very well across different antibody concentrations. Therefore, substrates with a midpoint in sensitivity, such as Super Signal West Dura substrate, shows more flexibility with different concentrations of both primary and secondary antibodies with reasonable exposure time for both high and low abundance targets. For data acquisition, our eye-bright imaging systems makes image acquisition and data analysis very easy with a variety of smart features, including advanced auto-exposure, automatic sample alignment, automated analysis, including quantitation and normalization, which can be performed on board the instrument. The eye-bright imaging system is capable of multiplexing up to 4 colors simultaneously from both visible and near infrared, and even chemi on the same image. The eye-bright imaging system allows you to achieve high sensitivity with an improved dynamic range. And speaking of dynamic range, this is also a consideration when performing quantitative Western blotting. When choosing your image capture method, note that both film and imaging systems will saturate when the exposure time is too long. Film, however, has significantly less dynamic range compared to an imaging system, and it will not tell you when saturation has occurred. On the other hand, imaging systems, they have a wider dynamic range and may have advanced algorithms to determine when saturation has occurred. As you can see in the plot on the right, with the smart exposure technology of the eye-bright imager, it automatically determines the best exposure time that is within the imager's dynamic range with no saturation. And if you see the manually set exposure times, the eye-bright is also able to determine that saturation has occurred, shown by the bands in red. Now, some users who are very used to the film method may argue that film has the best sensitivity. Here is some reference luminometer plate images captured on X-ray film and eye-bright imaging system, and you can see that the eye-bright imaging system is able to give much better sensitivity than X-ray film at all exposure times. Another consideration when performing quantitative Western blotting is protein normalization. Scientific publishers and funding agencies are now requiring other forms of normalization or validation of normalization controls to ensure reproducibility and accuracy of quantitative Western blotting results. Housekeeping proteins are the most commonly used normalization control. In the table on the right, you can see some commonly used housekeeping proteins such as actin, GAPDH, and tubulin. Housekeeping proteins are assumed to be expressed constitutively at the same levels across experiments. However, to reliably use housekeeping proteins for normalization, it is necessary to validate that firstly, your housekeeping protein is not being regulated by the experimental condition, and secondly, that your housekeeping protein is within the linear range and not saturated. The alternative to normalizing to housekeeping proteins is to perform total protein normalization. With total protein normalization, the abundance of your target is normalized to the total amount of protein in each lane, instead of being dependent on a single loading control protein. This method is sensitive. You can use as little as 0.1 micrograms of total protein per lane. And more importantly, it improves data reliability of quantitative Western blots. One final tip for the immunodetection portion is to check your transfer consistency before going on to immunoprobing. Doing this will ensure that you do not waste time and antibodies on a bad blot. You can check transfer either by labeling proteins or by staining them. Our in vitrogen no-stain protein labeling reagent labels the proteins, which can later be used to perform total protein normalization. The labeling takes only 10 minutes, is highly sensitive, and the signal is stable for years. The alternative is to stain the proteins, and for this, we have the Pierce reversible protein staining kit. However, staining takes slightly longer and is also slightly less sensitive. And just a note that for those doing fluorescence Western blotting, we recommend against using Ponceau S for staining, as even after washing off, it leaves a background stain on all bands in several fluorescent channels. When checking transfer, some commonly used tests is to check how much protein is remaining on the gel and how much protein has been transferred to the membrane. However, note that not all tests are indicative of good Western results. In the wet transfer on the left, you can see there is little protein left on the gel, and most, including the very high molecular weight bands, are transferred onto the membrane. However, after the blot is processed, many bands of interest are missing. On the other hand, with the fast dry transfer using our iBlot 2 system, you still see a lot of protein left on the gel post-transfer, and a good amount is transferred to the membrane. However, when the blot is processed, you are able to see all the bands of interest nicely. So what this means is a clean gel does not always mean good results. When a gel looks very clean, it also means that significant overtransfer could occur. Additionally, overtransfer may cause protein to bind weakly to the membrane, such that it is partially removed in immunoprobing. And as mentioned before, protein stacking on the membrane can affect linearity. So the bottom line is the protein amount, membrane, transfer time and voltage, and buffers used can have an impact on how well a protein binds to a membrane. This can influence if anything washes off the membrane in immunoprobing. Our iBlot 2 fast dry transfer system has optimized protocols to ensure that the most efficient transfer of your molecular weights of interest without overtransfer. In summary, in order to achieve reliable quantitative data from the Western blots and to avoid experimental variations, optimization, controls, and normalization are required to prove the accuracy of your results. This can be achieved by using a low gel load amount to prevent membrane saturation, confirming transfer consistency by membrane labeling or staining, normalization using housekeeping genes or total protein, and ensuring no image saturation by using an imager with a high dynamic range. At Thermo Fisher Scientific, we have a wide library of educational resources at our Protein Gel Electrophoresis and Western Blotting Education Center to help you with every aspect of Western blot analysis. And these include how-to videos, tips and tricks, on-demand videos, and our latest technologies, and also protocols. And these can all be accessed at our webpage, thermofisher.com/Western Education. So that brings me to the end of my presentation. Thank you very much for your time, and I will hand the time back to you, Megan.
Thank you, Dr. Audrey and Anthony Lim, for your informative presentation. Before we go to our live Q&A, we would like to draw your attention to the left side of your screen, where you will find a survey on today's webinar. We will now allow a few minutes for you to fill out that survey before getting to the Q&A. Thank you. Thank you all so much for taking the time to complete the survey. We will now begin our Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box and click Send. We'll answer as many questions as we have time for. Let's get started. Our first question is for you, Anthony. What are the pros and cons of using fluorescent conjugated secondary versus enzyme conjugated secondary antibodies?
Thanks, Megan. Thanks for the question. So fluorescent conjugated secondary antibody allows the multiplexing technique to be involved here. And in this case here, they are commonly used in techniques such as flow cytometry, immunofluorescence, immunocytochemistry, cellular imaging. Furthermore, fluorescent dyes typically have wider dynamic range as well, which allows more quantitative analysis. In reverse, the enzyme-linked secondary antibody allows colorimetric as well as chemiluminescence detection of the primary antibody, and they are widely used in techniques such as Western blotting, immunoassays, including ELISAs.
Great, thank you so much. Audrey, my next question is for you. How do the labeling reagents / stains work? Do they bind to the proteins? And if so, are they compatible with their downstream steps, such as blocking, antibody binding, etc.?
So that's a great question. Yeah, so I went through labeling as well as staining earlier. So labeling, like in the context of the no-stain reagent that I mentioned that we have for total protein normalization. So how that works is that it actually forms covalent bonds with the lysine amino acid residues, which is the most abundant in protein. So these covalent bonds, they're actually permanent. So that means that, you know, for all the downstream steps, it doesn't get washed off throughout the immunoprobing. Yeah, so that actually allows you to do the total protein normalization later on, after all the antibody incubation. On the other hand, staining, it actually stains the proteins, but that is also reversible. Okay, so what this means is that, yeah, usually you will wash it off before going on to immunoprobing. Yeah, but even then, with the immunoprobing, the blocking and the antibody steps and all that, it will get washed off. So the membrane stain, the staining, protein staining, is really mainly for checking the transfer efficiency, whereas for the labeling, it can be used for downstream total protein normalization, as the signal, you know, it stays on permanently. Yeah.
Great, thank you so much. And just as a reminder to our audience, if you have any questions to ask, this will now be your time to do so. But that brings me to my next question, which is for you, Anthony. Can you further elaborate on what is advanced verified antibodies? How is that different with non-varied antibodies?
Great question here. Thanks. So just to elaborate a bit further, in Thermo Fisher Scientific, our antibodies typically undergoes a rigorous two-part testing approach. So on the first part, which is known as target specificity verification, this step will help to ensure that the antibody will bind to the correct target. In this case here, our antibody are being tested using at least one of the following methods, which I shared earlier, just to ensure proper functionality in the researcher's experiment. This will include techniques like knockout, knockdown of the protein, or independent antibody verification, which is like measuring the target expression by performing or using two different differentially raised antibody that recognizes the same protein target, cellular treatment, relative protein expression, neutralization, and so forth. So this is the first part. And on the second part, we perform functional application verification, where these tests are used to help to ensure that the antibody works in a particular application of interest. And this may include like Western blotting that we shared today, flow cytometry, chromatin immunoprecipitation, immunofluorescence imaging, as well as immunocytochemistry. So this is the two-step approach that we take within the company, just to ensure that the antibody that we provide are of the highest quality, to ensure that the researcher has the best tool in their hands. Thank you.
Thank you. Audrey, my next question is for you. Why is linear response important for quantitation in Western blotting?
So thank you for the question. Okay, so yes, so in my presentation, I keep emphasizing on this linear response in order to get accurate quantitative results in Western blotting. Yeah, because this linear response is really the whole basis of doing the quantitation in Western blotting, right? So the assumption is that as my protein load increases, right, my signal is going to increase as well. So if the response is not linear, it's saturating, right, then you're not going to get accurate results. You won't be able to tell if the gene expression is really different or not. And this is especially important for housekeeping proteins, right? Housekeeping proteins are usually very abundant and have a high tendency to saturate. So this is really something that you should bear in mind when doing the quantitative Western blotting, right? When you're trying to compare if one gene is more expressed than another, right? So that is why, in my presentation, I actually talk give a lot of tips on how to prevent that saturation, right? Like don't overload your proteins, very, very important. Don't overload your antibodies. Choose imager with a wide dynamic range, choose the right substrate, and so on. All of these tips are really, to ensure the linearity of that signal.
Great, thank you. And it looks like we have one more question here, Audrey. What are some pharmaceutical industrial applications of Western blotting?
Yeah. Okay, so in terms of industrial applications, so like if, let's say in the pharmaceutical industry, so a lot of times they're actually synthesizing proteins, right? So Western blotting is one way to verify that they are indeed getting their protein of interest, right? And if, downstream, they're also looking at, how this protein is going to, in the R&D phase, right? How this protein is going to affect the entire workflow response, right, downstream, then Western blotting can actually also give an indication of what is being upregulated, downregulated, and so on.
Great, thank you. And another question we have here, as you mentioned, Audrey, that the labeling reagent works with covalent bonds with lysine amino acids. Is there a minimum number of lysine amino acids a protein needs to have for the labeling reagent to work?
Yeah, that's a great question. Yeah, so the reason why, when our R&D team developed this no-stain reagent and why we chose lysine is because we know that lysine is one of the most abundant amino acids. So with this in mind, you don't really need to be so concerned that whether there is a minimum number of lysine amino acids in your protein, because it is most likely going to be the most abundant one there. Yeah, so on the other hand, if you are working with other types of labeling reagents that maybe label other types of amino acid, then you have to be concerned that, like for example, like tryptophan residues that are not so abundant. Yeah, then you have to check whether your protein has sufficient amount for it to be accurate for normalization.
Great. Thank you so much, Audrey. Thank you both so much. Do you have any final comments for our audience tonight?
I guess let me start by sharing that protein analysis is a widely used technique in the field of biologics, right? So a lot of the protein or antibodies which are being used currently are also therapeutic drugs as well, like the use of monoclonal antibody to treat cancer as well. So the ability to analyze, to make sure that your protein are intact, the protein are expressed at an appropriate level is a very good technique, and it's also a very good methodology to ensure that what goes into the patient are of good quality.
And for my side, I would say that, at Thermo Fisher Scientific, we are really committed to transform the way you perform Western blotting to improve your results, get your results in a much faster time. Yeah, so do not hesitate to, reach out to your nearest Thermo Fisher rep. Yeah, so we'll be very happy to share with you, what are the latest technologies that we have that can improve your results.
Great. Thank you again, Dr. Audrey Lim and Dr. Anthony Lim, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.